So my topic is a bit different. I work in a very highly specialized field of medicine, inborn errors of metabolism. And we've worked very hard to adapt high-throughput sequencing of DNA into the clinic for individual patients in this very specialized field. So I work at the Karolinska Institute, which is our medical university, and I have a clinic at the Karolinska University Hospital, which is connected to our university. So, massively parallel DNA sequencing, high-throughput genetics, big data. This has, of course, changed very much how we can do genetics in medicine, and mostly this has changed things very much for clinical science, because we can do a lot of sequencing of complete genomes at a reasonable cost and rather quickly. So lots of cohorts of patients are being sequenced, and we can find risk genes for those disorders. But the challenge is, of course, to understand, not only manage the data, but actually understand what it means for an individual patient. And there are a few fields of medicine where this is possible today. And it's not really so much in the complex disorders like diabetes, which we called previously, rather in the inherited disorders that are more specifically genetic. And the type of disorders that I work with are monogenic disorders, single gene traits, whereas diabetes and the common diseases would be in the far right group there, which are extremely interesting to work with on a large scale, but not quite in the clinic for the individual patient yet. So monogenic disorders, there are about 10,000 known, and the incidence of these is really rising now that we can finally find them. And the proportion of the population affected would be approaching 10%. And the group of monogenic that I work with then affect metabolism. So this is a situation, this is a family, two parents having two children that both die, in this case, from a severe disorder that affects metabolism. And up to now, it has been, in many cases, impossible to know exactly which gene out of all 25,000 genes has been responsible. So we have to tell our patients that you have a 25% risk each time you have a baby that the same thing will happen. And this is where we can now make a major impact. So this is intermediary metabolism, all the myriads of chemical reactions that take place in all our cells continuously. And each of these reactions have a gene responsible. And so there are thousands of disorders, and each of them are rare, but together they are a significant disease burden. And the symptoms of these disorders are either that you accumulate toxic metabolites, or that you cannot produce a vital product that you need, and that product could be energy. So as clinicians, we deal with a range of different types of patients, which is very challenging. A classical form can be the newborn baby who's born healthy because you're protected in the womb by your mother's metabolism, but then when you're on your own, you become critically ill. That is one presentation. You can also have a very gradual, progressive presentation. And most patients will be mentally retarded unless you find them in time. That is a common thing with all these patients. But they can also be adult. We have lots of patients who become ill in adult life. And it can be, for instance, the mitochondrial disorders. They can affect any organ or any combination of organs. So as you can understand, diagnostics is very challenging. We have thousands of disorders, each are very rare, and they turn up across all clinical disciplines. Now, many of these diseases can be treated if we know what's wrong. We can take away a compound that you cannot metabolize, or we can add something that you can't make. So diagnostics is important. And since the brain is affected, we need to be very quick and very early. So that's the challenge. So to meet this challenge, we have a clinic. I head this clinic, which is called the Center for Inherited Metabolic Diseases. And we have then implemented, as I will just tell you now, the new genetics. And what's crucial for our success here is the multidisciplinary way of working. So we worked very hard to put together a team containing all the relevant expertise: clinicians, pediatricians, neurologists, endocrinologists, clinical geneticists, that's me actually, clinical chemistry, biochemistry, analytical chemistry, and genetics, bioinformatics, and mathematics, actually. So we sit together handling these large amounts of data. What's important is that we have a very long experience with covering these disorders because we are the National Center for the Neonatal Screening Program in Sweden since 1965, actually. So we have the networks across the country for rapid action on rare diseases, which is ideal for us when we go into the next generation genetics. So what we have done, we have worked very intimately with the Science for Life laboratory, which is a national resource that was funded by the government a few years back for high-throughput biology. And this is mainly a research institute. But we have managed to establish a clinical platform that we are working very closely with. And this is a pilot project with these disorders. So we do the whole exome, which is all protein coding parts of the gene, or actually we're now moving into doing the whole genome in patients, which is, of course, big data, a lot of information. But the critical thing here is that we don't look at the whole genome, but from that source we extract only the clinically relevant information and only the information that we have full coverage in our expertise for rapid action. Because ethically we don't want to know if these small babies have inherited risk of breast cancer, for instance. So we've spent a lot of time developing tools to only filter out the relevant information that we can act upon. And we are establishing this so other clinical teams can take on other disease areas. And I will show you then what types of tools these are. But I will only just say that this has also the potential for science, because in the patients that are not explained but have very typical metabolic disorders, that is where we make the discoveries of new disorders. So in the clinical track, we are taking a very big step forwards in what we can do for our patients. And in the scientific track, we are making real discoveries and finally understanding how these disorders arise, which is very important as well. But they are very different, because in the clinical track, you need to be quick. And in the scientific track, you need to be very thorough and have proper informed consent, etc. So those are very different, but very much related to each other anyway. So the tools that we have developed is first one to call this clinical grade, because it's lots of genetics done on patients which is not really meeting clinical standards. One is to segregate the data or filter the data so we can only see the relevant parts, filter in the families, and also score all the variants, because we have lots of variants in our genomes. Score them according to disease-causing potential. We made a program for quality, which is not really available anywhere else because people are so anxious to get results. But we made a program so we can, for each base in the genome, we can exactly see the quality and amount of coverage. So we can evaluate a negative result, which is very important. And we made a web-based interface for visualization, so our clinicians can sit and see only the relevant data scored for pathogenicity. So it takes about 15 minutes to go through one patient. And then we have a database, so we only go through those genes that are validated, proven causes of disease in our area where we have competence, which is very, very important. And then finally, we have the clinical team. And we made this pipeline so we can exchange the database of genes with any other list of genes, provided that there are clinical teams specializing in other disease areas. So this is generic. So to sum up, this has been a dramatic clinical impact. We're now diagnosing so much more patients. Among these families with unclear symptoms, children with encephalopathies that are not developing normally, we can now find so many more monogenic disorders. It's really a quantum leap in what we can do when we find them before they are irreversibly damaged, finally. And this test is very stringent, so we're not doing the genome in patients. We are filtering out only the relevant parts of the genome. The rest is still research. And it's really, we were very enthusiastic when we started, but it's still overwhelming to see what is emerging because it's a totally new landscape. So many different new disorders that maybe there are 2 or 3 patients known for each of them. They are now emerging, and now for the first time we can start treating them before they are actually ill. So all the clinical science is now coming into place, not only the genetics, but actually putting in the treatment at the right time and evaluates how the treatment actually works. Regular old-fashioned clinical studies of these patients. And of course, there is a very strong scientific component here because we're finding new causes of disease, new mechanisms, and going into model system and trying to find new treatments for these patients based on that. And last point is that these disorders are, of course, a way into understanding common diseases, because we know, for instance, for mitochondrial disorders, that there are links between mitochondrial dysfunction and neurodegeneration, but we don't know the mechanisms. And these sort of human monogenic models are a way to address those problems as well. So many people involved, my clinic, the university, and the Science for Life laboratory, basically. Thank you very much.
Thank you very much, Anna. Thank you. One very quick question, because I mean, I know you mentioned it in the end, but I mean the whole ethical aspect, because I know that you are, you know, you're emphasizing the possibility of early cure, which is, you know, the big possibility here.
Yes.
But you know, there's a different flip side of that coin as well, where people can say, well, if we can track this early, you know, we could do, you know, preventive other stuff early. I mean, where do you feel that, you know, there's the border between, you know, the ethical border there?
Well, in these cases, it's not very hard, because the downside of not finding these babies early is very obvious, because that's what we have today. We have so many patients without diagnosis, which are terrible cost in both, you know, human suffering and money. So in this field of medicine, it's not so difficult. But I think the key is the multidisciplinary team, because they have to make the decision on when patients should go into this track, and it shouldn't be done without knowing what you're looking for and taking responsibility for that. So in this area, I think it's pretty straightforward.
But there is an aspect, and as I said, as food for thought, you know, it's something to bear in mind as well, you know, when it comes to the whole being able to track everything also leads to, you know, with great powers comes great responsibility.
Yes. Now, we think this is ideologically important, because many people do a lot of whole genome sequencing under very much less stringent conditions. So this is the way we would like to advocate. Focus on the disease area where you have competence, and put together the multidisciplinary team and do that, and let other experts do the other stuff where they have the knowledge.
Thank you very much, Anna.
Thank you.
Thank you.